All right. Hey, anatomy and physiology students. This should be a relatively quick recording. I'm just going back and reviewing the practice questions that we went over for this chem and cell bio activity that we did last Thursday, January 11th. So I have made a copy of the slide deck that you guys were working on and have made some additions to it and edits to it based on trying to add as much info as possible that you could have included for these, as well as correcting any misinformation. So these edits are all in red when I pull out those slides, and you won't see these on the copy that you guys have access to. So if you would like to update your slides with the information that I have added here, you're going to have to do that yourself. So the whole purpose is to get you to spend more time with the content in that way, rather than me just writing down the information that you already have in the other slides, right? So at the very least, if you're going to use the exact same wording that I used here, at least you will be typing it, and that should help form some sort of memory formation with the info. All right. Anyways, let's get into this. So I set up slides with a few example open-ended questions that are similar to what you might see on the Unit 1 exam, right, to give you practice thinking about and analyzing this content. So question 1 was addressing bonds. So I asked you to describe each of the bond types that we talked about and give an example of a type of molecule or an example molecule that is formed with each bond. And so our main atomic bonds, so those that are forming bonds between individual atoms, are covalent bonds and ionic bonds. And within covalent bonds, we have two different types. We have polar and nonpolar. But essentially, the main difference between a covalent and an ionic bond is that a covalent bond includes the sharing of valence electrons, whereas an ionic bond is going to form from the exchange of valence electrons. So typically, a covalent bond will have shared pairs of electrons to fill the valence shell of all of the elements involved. And then an ionic bond is going to, with that exchange of electrons, one of the elements is going to lose its valence shell, essentially moving down in energy level to its full shell below it. And then the other element, the more electronegative element, is going to gain the last electron that it needed. So typically, an ionic bond is formed between an element that only has one valence electron and an element that has 7. So the larger element, or the element that has 7, only needs one more, and the element that has 1 just needs to get rid of that electron in order to now have a full shell. It would not be able to fill its shell through covalent bonds because it only has 1 electron to form a pair. Hopefully that all makes sense. So anyways, asked to address polar versus nonpolar, right? So covalent bonds, in both examples here, we have electrons being shared. However, not every electron sharing is going to occur equally. And so that's how we get polar covalent bonds, when the sharing of electrons is unequal. So a polar covalent bond is when atoms share valence electrons unequally, creating poles, essentially. That's the name polar. And so anytime a pole is formed, there's going to be a positive and a negative side. So if you think about a magnet has a positive pole and a negative pole. Even if you snap that magnet in half, now both of those magnets have a positive and a negative pole. There's always polarity in those types of examples. So for our example here, we have water, H2O, which is going to be our most common polar covalent molecule that we discuss. And so even though the oxygen and both hydrogens are sharing their electrons on their valence shells, oxygen is a much more electronegative element, and thus is going to have a stronger pull on those electrons. And so the electrons actually spend more of their time closer to the nucleus of oxygen than they do to hydrogen. And so you end up with 2 poles on this molecule. The side of the molecule that oxygen is on becomes slightly negatively charged due to the presence of those electrons being close to it. And then the side with the 2 hydrogens becomes a positively charged or slightly positively charged pole. So that gives water and other polar molecules the ability to make those magnetic attractions. So pulling on other nearby polar molecules or charged elements such as ions, creating what we call hydrogen bonds. So that slight attraction between positively and negatively charged molecules or elements creates those hydrogen bonds. And again, that's because oxygen is electronegative and so is greedy with those shared electrons. In a non-polar covalent bond, we have sharing of valence electrons, but those are shared evenly. So there is no net charge on any of the elements involved, right? We don't get any poles. And so that molecule is not charged. It is not going to have any interactions with other charged molecules in that way, including water. So it cannot interact with or dissolve in water. So like fats, oils, right, or lipids are mainly non-polar covalent bonds, or have mainly non-polar covalent bonds between the carbons in the fatty acid chain. And so they cannot bond with or interact with water. And that's why you get that clear separation. If you pour some oil into a cup of water, you can mix it up. And then after a few seconds, the oil will all separate from the water and create a film because it cannot bond, it cannot interact. Other common examples are anytime elements bond with other atoms of the same element. So like H2, O2, C2, right? Those are all going to be non-polar covalent bonds. And most of the carbon bonds in biological molecules, so in our carbohydrates, lipids, nucleic acids, proteins, as long as they are bonded with hydrogen, that is going to be a non-polar covalent bond, or if they're bonded with carbon. So it's typically only when you see side chains that include oxygen, nitrogen, phosphorus, that you end up with some charge or polarity to that molecule. And so lastly, we have our ionic bonds. And like I said, in an ionic bond, the less electronegative element is going to give away an electron to the more electronegative element. And so the first part of an ionic bond formation is that exchange. So an electron is given to one element, thus the element that lost its electron. So for example, we have sodium chloride here, NaCl, which is table salt. Sodium only has one valence electron, so it gives that valence electron to chlorine. Sodium is now left with one less electron than proton. Right, since it gave away one of its electrons. Thus, it is now carrying a full charge of +1. Right? It has one more proton in its nucleus than it has electrons in its cloud. And so it has become a charged ion, a charged element called a cation. Cations are positively charged. So cat has that T in it, that plus, for positively charged ions. The chlorine, in this example, which has now gained an extra electron, now has a full negative charge on that element, since it has one more electron in its cloud than it does protons in the nucleus. And so that has become an anion, a negatively charged ion. And so now that we have two ions with opposite charges, that is going to cause that electrostatic attraction, which forms our bond here. Cool. So those are our types of bonds. And so kind of expanding on the idea of polar molecules and how they interact with other molecules, this question is asking, how does water dissolve ions? And since water is polar, it can interact with either positively or negatively charged elements. And so whenever ions or polar molecules are added to an aqueous solution, aqueous meaning mainly water-based, water is going to start bonding with it and separate portions of the molecule. And so specifically with salts, which are ions, the positively charged or slightly positively charged hydrogens in the water molecule are going to surround the anion, and then the slightly negatively charged oxygens in that water molecule are going to surround the cation. And so this forms a sphere, right, like a boundary, a barrier of water molecules around each ion that we refer to as hydration spheres. And so it is those hydrogen bonds, those slight attractions between the polarity of water and then the charges of the ions that attracts them to each other, pulls them apart, and then creates these hydration spheres, keeping them separate. Okay? If you dissolve salts in water, those ions are going to stay separate from each other unless you evaporate all of the water, thus leaving space for those ions to bond again. Okay. Question 3 was asking you about the protein fibers that make up the cytoskeleton. And so we talked about the three major protein fibers, the three examples. So we have actin filaments, tubulin filaments, and intermediate filaments. So they're all proteins that have a slightly different formation and thus slightly different function. So our actin filaments, aka microfilaments, are the smallest or thinnest version of these protein fibers. They're responsible for a variety of cellular processes that typically are involved with some sort of support as far as the shape or movement of that cell. Actin fibers are kind of like a twisted rope, And so they have the ability to extend and then contract, and thus they also have the ability to generate force. And so actin filaments are what make up the contractile portions of our muscle. Actin filaments are what are used in the extensions of the plasma membrane that have slight movement, such as, that don't have movement, such as microvilli. Okay. And then our microtubules are the ones that are included in those plasma membrane extensions that do move. So that's our next one. Tubulin is the protein that makes up microtubules. So these are our largest cytoskeletal protein fibers. And tubulin is mainly there for structure and support. They create the actual skeleton portion of the cytoskeleton. They easily and quickly go through polymerization and depolymerization, so extension and then shortening. Our cells have the ability to quickly add on individual tubulin dimers, whereas the actin filament is kind of that twisted rope of those actin proteins. Tubulin is like a bunch of little balls that are added onto each other and twist and twist and twist, and it creates a tube, thus the name. It is a hollow cylindrical tube that creates these microtubules. And so since these tubules can quickly and easily lengthen and then shorten, they're used often in the movement of molecules. So we talked about during cellular replication, the extension of those spindle fibers that connect with sister chromatids and then pull those sister chromatids towards each pole of the cell to create our new nuclei. That is the polymerization of tubulin and then depolymerization to pull back. Some of the more permanent tubulin structures act as tracks for other proteins to move along, such as kinesin. There's that video that we shared with that kinesin molecule moving along the tubulin track. And then, like I said at the beginning, they make up the plasma membrane extensions that are responsible for movement or do move a cell, such as cilia, which in most of our human cells, our cilia are there to move molecules across the membrane. They do this power stroke motion, but other cells, other organisms like bacteria can use cilia for movement, and then flagella. So the only cell in the human body that has a flagella is the sperm cell, uses the flagella to move. Right? But other than that, we don't have any flagella, though they are more common in bacterial cells, again, as movement. And then lastly, we have our intermediate filaments. So as the name suggests, these are the intermediate size, the medium size. So we have our microfilaments, which are smallest, our microtubules, which are largest, and then our intermediate filaments. And these kind of make up other structural components that are more permanent and less variable in how they function. They're part of a lot of the interconnecting webs of cells in our extracellular matrix. So they can make sheets, they can keep the cell together and neighboring cells from disconnecting. And keratin is a very common protein-specific example of protein that we'll talk about a lot in this class. Keratin is an intermediate filament, and that's what makes up the hair and nails in our bodies, makes up scales and feathers in other organisms. So yeah, those are our various cytoskeletal proteins, fibers, proteins. Okay, question 4 and 5 are then analyzing the major macromolecules in living organisms. So for each, I'm asking you to identify their functions and then address the elements that make up those types of molecules and then their relative ratios when applicable. I also gave the option to insert images here, but on these slides, it can take up a bit too much space, though it does look like we have some on the next one. So for carbohydrates and lipids, both elements are composed of carbon, hydrogen, and oxygen only. You will only find those 3 elements in a carbohydrate or a lipid. Carbohydrates will always have these elements in a 1 to 2 to 1 ratio. So for every 1 carbon, there is 2 hydrogens and 1 oxygen. Lipids have carbon and hydrogen in that 1 to 2 ratio. So for every 1 carbon, there are 2 hydrogens. However, there is much less oxygen compared to carbs because of what lipids are composed of. Lipids have a glycerol head, which will have an oxygen or 2 on that head. That glycerol head is technically a small carbohydrate. But then a majority of the mass of that lipid is made up of the fatty acid tails, which are literally just long chains of carbon and hydrogen. Okay, sorry, I got a phone call and had to pause. I believe I left off talking about the fatty acid chains of lipids, right, which is mainly composed of the carbon backbone and then hydrogens bonded with those carbons. So a majority of the mass of the lipids is made up of carbons and hydrogens, with just a couple or a few oxygens attached to that glycerol head. So as far as functions of these molecules, carbohydrates, their primary function is to act as our cell's primary source of energy, especially during activity. So there are some cells of the body that can only use carbohydrates or very preferentially use carbohydrates. So our red blood cells, which do not have a nucleus or a mitochondria, only can use carbohydrates as an energy source. And then our neurons, the nervous cells that make up a majority of our nervous system, or make up our nervous system, mainly will use carbohydrates, though they can use others as a source of energy. So yeah, carbohydrates' primary function is as our source of energy during activity for a majority of the cells in our body. Carbohydrates also serve as or perform structural functions for cells, especially in like plant cells with cell walls and bacteria with cell walls. But we also have structural carbohydrates such as like glycolipids and glycoproteins, which also help with communication between cells as well. So lipids, aka our fats and oils, are used for lots of different things too. So they also provide energy to our cells. They are our primary source of energy at rest for a majority of the cells in our body because they are much more energy dense than a carbohydrate. Our cells aren't as efficient at breaking them down. So like carbs provide efficient energy fast when we need it. Lipids provide more energy. It just takes a little bit longer to transform them and then break them apart. Lipids also provide insulation, storage for things like lipid-soluble vitamins, protection as part of our epithelial lining in between a lot of our organs, and they make up the majority of the mass of our cell membranes, right, as phospholipids. Right. And so continuing on with our macromolecules, we have proteins and nucleic acids. So both contain carbon, hydrogen, and oxygen, but they also have a couple other elements, such as nitrogen in our nucleic acids, and then we can have nitrogen, sulfur, and phosphorus in our amino acids, which make up our proteins. Proteins essentially are the tools of the cell. They do everything, right? They are what are carrying out the instructions that are contained within our genetic material, within our nucleic acids. And so they do everything, right? Besides being a primary energy source, they are our last go-to as far as a source of energy. So we only break them down if our bodies really need it and we're out of glucose and lipids. But other than that, they do everything: transport, structure, etc. Nucleic acids are what make up our genetic material. So all of our DNA and our RNA. They are made up of a nitrogenous base, a pentose sugar, and then a phosphate group, like you see pictured here. Whereas amino acids they have an amine group, but they also have that variable R group, which changes between each individual amino acid. All of our nucleic acids will have this structure, and it is the nitrogenous base that has slight differences between all of our nucleotides. So A, T, C, G, and then U in the RNA. Right. Question 6 is asking about pH and buffers. So pH, you guys did a great job here. pH is a measure of the acidity or basicness of a solution, and it is based off of the concentration of free protons, so these positively charged hydrogen ions. We calculate the pH of a solution by taking the negative log of the concentration or molarity. Molarity is a term for concentration of those protons, so those hydrogen cations. It's just a proton. So essentially at the most basic level, an acid is any solution that it freely gives up protons. And then a base is any solution that accepts protons, so takes free protons out of that solution. And so our pH scale goes from 0 to 14, or 1 to 14, with 1 being very acidic, 14 being very basic, and then 7 being our neutral. So water, H2O, is a 7. You can split water apart. If you take a proton off of that water molecule, you have a proton and a hydroxyl group. The proton would be the acid, the hydroxyl group would be the base, and so it is also a perfect buffer in that way. A buffer is an aqueous solution that can either resist changes in pH levels or actively neutralize the pH of a solution. So just resistant to those changes in pH in both directions. Question 7 here, I gave you a picture of a mitochondria, or mitochondrion is the singular for mitochondria, and just asked you to identify what is being labeled here. So the mitochondria has a unique structure compared to the other organelles in our animal cells. The only other organelle that has this similar structure and similar function is the chloroplast of plant cells. But the main difference or main identifying structure is that mitochondria have two membranes. So A is pointing to our outer membrane, B is pointing to our inner membrane. And since we have two membranes, we also have further compartmentalization of the space in this organelle. So we have two different spaces. We have the space all the way inside the inner membrane called the matrix, and then the space in between our inner and outer membranes called the intermembranous space. And so if you remember from talking about cellular respiration, we have our three main phases or stages of cellular respiration. Glycolysis occurs in the cytoplasm outside of the mitochondria. And then if oxygen is present, the substrates, the pyruvate from glycolysis will move into the mitochondria all the way into the matrix. The matrix is the site of the TCA cycle, which stands for the citric acid cycle, or tricarboxylic acid cycle, or it's also known as the Krebs cycle. I'm sorry, there's so many names for it, not my choice, but that cycle, the second stage, occurs in the matrix. And then the electron transport chain with ATP synthase, those proteins involved are embedded on the inner membrane. And so moving along that same idea, the next question was asking how the electron transport chain functions, as well as ATP synthase in order to generate ATP. So we get much more efficient energy production, ATP production, when oxygen is present in our cells, and we can use our mitochondria to complete cellular respiration. Glycolysis only produces a net of 2 ATP, and if no oxygen is present, the other cells in our body are actually going to have to utilize energy to help recycle some of the things that get produced, right, such as lactic acid. So anyways, when oxygen is present, and pyruvate can move into the mitochondria and go through the TCA cycle, through the electron transport chain, we make much more ATP. And the way this works is that each of those two previous phases, glycolysis and the TCA cycle, are going to transfer some of the energy from that glucose, from that original glucose molecule, as electrons, as high-powered electrons that are carried by some electron carriers. So these are NADH and FADH2. Those electron carriers will carry the electrons all the way to the inner membrane of the mitochondria and donate the electrons to cytochrome proteins along the inner membrane. These cytochrome proteins are what make up the electron transport chain, and they will essentially move electrons from one protein to the next along a chain. And those electrons are being drawn along that chain by oxygen. So oxygen, again, being very electronegative, is going to help pull the electrons down this chain, and ultimately those electrons will end up on oxygen and create water. So every time an electron moves from one cytochrome to the next, that is going to cause a proton, so another hydrogen cation, right? So the proton to be pumped out of the matrix and into the intermembranous space. So we have a bunch of electron carriers in the matrix. They donate electrons to these cytochrome proteins along our inner membrane. And those proteins are going to pump protons out of the matrix and into this intermembranous space. And so by the end of all of our electron carriers donating their electrons and those electrons moving down the entire electron transport chain, we end up with a whole bunch of protons in that intermembranous space. So we get a really high concentration in the intermembranous space and a low concentration in the matrix, thus creating a strong gradient, right? Concentration gradient. And those protons want to move or diffuse to the matrix. They want to spread out. But the only way they are going to be able to cross that inner membrane is by traveling through a specialized protein called ATP synthase. And just as the name suggests, synthesis, the protein's role is to synthesize ATP, make ATP. And the flow of protons through that protein is what generates the kinetic energy to allow ATP synthase to shove a phosphate group onto a DP and make new ATP. That's what we get here, right? So the flow of protons through ATP synthase down their concentration gradient generates kinetic energy, causing the movement of that ATP synthase protein, which generates a whole bunch of ATP, much more ATP than the other two steps we're capable of producing, on average around 30. There is no exact number because energy is lost during every chemical reaction. Sometimes more energy is lost than others, but in general, around 30 from just this process, whereas we only got like 2 to 4 from the other two. But again, this requires oxygen to be present. That's really, really important. Neither the TCA cycle or the electron transport chain are going to occur if oxygen is not present. The mitochondria will not be used for energy production unless oxygen is there in order to draw electrons down the electron transport chain. Okay. Question 9 is addressing the structure and function of the portions of the plasma membrane. Or cellular membranes in general, right? So we have the plasma membrane, which is surrounding the entire cell, and then all of our organelles have their own cellular membranes. Vesicles, which are transporting molecules across the cell, have membranes. And so the general structure of those membranes is all the same. So they all have a phospholipid bilayer, which is 2 layers of phospholipids in opposite orientation. So the heads of these phospholipids are carrying a charge. They are hydrophilic, and so they will point towards the environment in both directions. So towards the external environment, which is almost always going to be an aqueous solution, and towards the internal environment of the cell, and the cytoplasm is aqueous. And so those heads allow for the interaction with that water. But then the tails, which make up the inner portion of this bilayer, are hydrophobic. And those hydrophobic tails prevent the diffusion of most large and or hydrophilic molecules. So anything or most molecules that are carrying a charge or that are polar in some way are not going to be able to freely diffuse through our membrane. And thus they're going to need help from something else to do that, such as these transport proteins, which we'll get back to in a second. Phospholipids are mainly liquid at our body temperature at 98.6 degrees. And so a different type of lipid, cholesterol, can be embedded within that bilayer in order to provide some stability, make it more solid in those regions. So we also have glycolipids and glycoproteins. So glyco referring to like glucose, sugars, and then lipids or proteins. So these are lipids and proteins that have carbohydrates attached to them. And they can be used for communication, as well as creating like adhesion molecules or adhesion interactions that hold neighboring cells together. And then lastly, we have our transport proteins. So these are proteins that are embedded into the membrane that either are creating channels for molecules to move through or will physically transfer molecules across that membrane. And so molecules that are going to be going against their concentration gradient or are especially large are going to require some energy in order to facilitate that movement. And usually that energy is going to come from ATP. So anytime ATP is providing energy for that movement, that is called active transport. And then anytime ATP is not required for the movement of those molecules, so if it's going with its concentration gradient, or if the movement of another molecule through that same protein is going with its concentration gradient, that can generate some energy. So whenever transport is occurring without the use of ATP, that is passive transport. Right. Recognizing the major or most common intracellular versus extracellular ions is going to become important later in this course when we start talking about the movement of ions to generate electrical potentials that cause the opening or closing of some other membrane proteins and the transduction of signals down membranes, such as with our muscles and neurons. And so in general, it's really important to recognize where we have higher concentrations of which types of ions. So inside of our cells, or yeah, wait, no, I didn't double check this. This should be opposite, actually. So let me go ahead and fix that. This should be extracellular. and this should be intracellular. So outside of our cells, we have sodium cations and chlorine anions in high concentrations. And then inside of our cells, we have potassium, phosphate, and then proteins, which aren't necessarily ions themselves, but do often carry a charge, acting as our intracellular ions. Right. And so, like I said, this is going to be important later when we start talking about the movements of ions to generate those potentials. So recognizing that sodium and chlorine are more common outside of the cell. And thus, if we are going to move them somewhere, we're typically going to be moving them inside the cell. That changes as well. So maybe that's not useful. Sodium and chlorine are more common outside of the cell. Potassium, phosphate are more common inside of the cell. And then proteins also play a role in the general charge of our membranes. Right. Question 11 is looking at osmolarity of solutions, specifically surrounding our cells, and the effect that would have on our cells. And so the most common example we talk about when looking at the effects of the osmolarity of a solution are our red blood cells. And so osmolarity is referring to the concentration of the solute that is inside versus outside of the cell. So essentially, we're still looking at concentration gradients. There's a high concentration somewhere and a low concentration somewhere else. It's going to cause or drive the movement of that molecule. But since we have membranes surrounding our cells, that often blocks the movement or causes other things to occur. So we have terms for the osmolarity of a solution that a cell is within. And so an isotonic solution is referring to even concentration on both sides of the membrane. So there is no net diffusion or osmosis in either direction. And they are constantly diffusing and water is diffusing. So osmosis is occurring in both directions constantly, but there's no net movement. It's not being driven in either direction. And so an isotonic solution for our blood is 0.9% sodium chloride, 0.9% salt. So that is an even concentration with the amount of salt that's inside of our cells. And so there's no net movements. A hypertonic solution has a higher concentration of a solute than what is inside the cell. And thus that is going to draw water out of the cell, which could cause the cell to shrink or crenate, like what you see here. A hypotonic solution has a lower concentration. These prefixes, hyper means above, hypo means below. So a hypotonic solution has a lower concentration than what is inside of the cell. And so that is going to draw water into the cell to try to create more space, which will cause the cell to grow in size and potentially even burst if the plasma membrane cannot hold all of that space, which is referred to as lysis or lysing. All right, and our last two slides here. So there is a lot that can be said about endocytosis, but really all I'm looking for you guys to recognize is what was included in this first paragraph. You guys did a great job here. So endocytosis is a form of active transport. It is the process of bringing substances into the cell by creating a vesicle. So instead of just a protein simply moving the molecule from outside to inside, this is going to involve receptor proteins that are going to bind with the ligand, so whatever solute molecule we are trying to move into the cell. And so those proteins with the ligand bondotome are going to stay embedded in the membrane. And the membrane will start to, I'm trying to think of the best way to show this. This membrane will start to kind of fold in. Okay, so if our receptors are in here with the ligands, the membrane will start to fold in until eventually it meets and pinches off completely. And so the plasma membrane surrounding the cell never created an opening. It just folded in, pinched together, and now we have a fully surrounded vesicle with a membrane with all of whatever we're bringing in, the ligand, in the center. And so that is endocytosis. So cytosis is cell movement, endo is into, so moving into the cell, endocytosis. It creates those membrane-bound vesicles, so keeps whatever we're bringing into the cell separate from the cytoplasm. And essentially it's an active process. It requires ATP, and that's what I want you to recognize. We have a few different examples, but really just want that general process of how endocytosis works, and how it's different than other examples of active transport where the molecule is being moved directly into the cytoplasm of the cell. And I guess expanding on this, exocytosis is generally the same process, just in reverse. So a vesicle fuses with the plasma membrane and then opens up into the exterior, releasing whatever ligand or molecule is contained within it. Lastly here, we have protein synthesis, or the creation of proteins and the various stages that go into it. So this is referring to the central dogma of biology, the core of how all living things function, which is the expression of the instructions that are contained within our genes, within our DNA. And those will always lead to the production of a protein that will carry out those instructions. And so there are two main processes in protein formation and protein synthesis. We have transcription, which is creating a copy of DNA, a transcript out of RNA, specifically messenger RNA. And then we have translation, which is the process of reading that transcript and turning it into a protein. So transcription occurs within the nucleus of our cells. So whatever gene we are reading will unravel from its nucleosome and then completely unravel the double-stranded molecule so that we can read the template strand. And RNA polymerase is then going to start reading that nucleotide strand and create complementary messenger RNA. And so by the end of transcription, we have a transcript of just that segment of DNA, just that segment of our gene that is going to create a single protein. That mRNA transcript can then leave the nucleus and go to a ribosome. And that ribosome will then read that mRNA transcript by translating 3 nucleotides at a time. So each nucleotide, or each functional unit of that gene, which is going to produce 1 amino acid, is called a codon. Codons are 3 nucleotides that code for 1 amino acid. So every 3 nucleotides is going to correlate with 1 amino acid. That amino acid will be brought by transfer RNA, tRNA, and then that will start to create a bond with the next amino acid. So the ribosome just moves down the transcript, reading 1 codon at a time, adding 1 amino acid at a time to this extending polypeptide chain. And by the end of the process of translation, we have created a linear chain of amino acids. And so that is the primary structure of a protein, right? Just that linear polypeptide chain that is released from the ribosome after translation. That polypeptide chain will then start to form interactions with itself based on the polarity of some of the amino acids, creating hydrogen bonds that are going to create that secondary structure, which is either a alpha helix or a beta sheet, pleated sheet. And then within the endoplasmic reticulum, and especially within the Golgi apparatus, we get further protein maturation, which is going to have other proteins help fold that secondary structure into its actual functional form, which will be the tertiary or quaternary structure. So a single polypeptide in its functional form is in its tertiary structure, but then some proteins are multiple polypeptide chains that are bonded together, which is what creates quaternary structure. And so then that protein will be transported out of the Golgi apparatus to wherever it's going to function. And those are essentially the stages of protein synthesis. So hopefully all of that made sense. Again, you're very likely to see at least a couple, if not a few, of these open-ended type questions asking you to explain a process in as much detail as possible, where it's absolutely possible to earn partial credit, but I'm looking for you to exhibit a full understanding and as close as you can to this kind of thing. I'll have, for my key and how I grade, is going to be based off of core details that should be included when addressing them. But yeah, so expect at least a couple questions like this. If you have questions or concerns, please reach out to me. All of this info should be available in the PowerPoint slides, and we should have talked about it in class. So yeah, you have those resources as well to review. So yeah, that's all for now. See you guys in class.